Welcome to our latest installment of the Spark Seminar Series. I'm Sue Tappan. I'm a member of the Spark DRC. Please join me in welcoming Dr. Don Bolser from the University of Florida. Dr. Bolser's research approach combines advanced computational neural network modeling with large multi-neuron in vivo recordings. These predictive models can inform therapeutic approaches for patients with neurological diseases affecting cough and swallow function. He has significantly contributed to the evidence-based clinical practice, too, including his work on the consensus panel report, Managing Cough as a Defense Mechanism and as a Symptom. Today, we'll be hearing about his current research involving brainstem circuits for reflexive control of breathing. As a Spark-funded researcher, Don has shared data and knowledge on the Spark portal, and you can too. Spark connects the body and the brain with open resources that support research into systems physiology and neural control in the periphery, such as our computational platform OSpark and the annotated maps which are generated from SCAN, our knowledge base of peripheral connectivity. Spark is a member of the AWS Open Data Sponsorship Program, which enables all public datasets to be stored and accessed without cost. Spark ensures interoperability across diverse contributions to advance basic science, clinical applications, and bioelectronic devices. So without further ado, Dr. Bolser has transformed our understanding of how the intricate neural circuitry underlie the vital reflexes that coordinate breathing, cough, and swallow. I'm looking forward to this deep dive into the brainstem, Don. The floor is yours.
Well, thanks again, Sue, for inviting me to share some of our work with you, and the work you'll see, some of which is funded by an OT2 grant through Spark, but also several other R01s. And the work is a collaborative process between two teams, primarily here and the University of South Florida, but also our collaborator, Ivan Poliacek from Kamenetsky University in Slovakia was an important contributor as well. So it's a team science project and has a lot of moving parts. But and I'm thankful to everyone that you see here listed, as well as funding sources, including that from Spark, which was a real shot in the arm for this kind of work. As you can see, we're going to talk about central circuits mediating sensory feedback from the superior laryngeal nerve, which leaves the larynx and enters the vagal trunk around the nodose ganglion. And in particular, I'm going to focus on a single behavior known as the laryngeal adductor reflex. And I'll talk about what it is, but it's a behavior that's actuated every day by otolaryngologists as a way to understand the competency of the function of the larynx. And typically what happens is the vocal folds close and prevent intrusion of material from the pharynx into the airways. Okay, so it's an important what we call airway defensive behavior or airway protective behavior. And so let's see. So when those airway protective behaviors like that one break down, the consequences are severe, including aspiration, pneumonia, but also other sorts of issues, nutrition, dehydration, and typically that's associated with disordered swallowing, or dysphagia, and often severe dysphagia is associated with impaired coughing called dystusia. And there's a high mortality when there's a severe breakdowns in airway protective mechanisms simply because of recurrent pulmonary infection. And so there's this suite of behaviors that the brainstem typically mediates, starting with cough, swallow, augmented breath, these other behaviors you may not be familiar with. And also, finally, the laryngeal adductor reflex. And each behavior has its own sort of control system in the brainstem, and there's a lot of overlap. I'm focusing on the laryngeal adductor reflex because it's a straightforward behavior, and one might think that its control system is fairly straightforward also, and that's the main hypothesis, but it's actually a lot more complicated than that, right, as you'll see. So an example of what happens when airway protection breaks down is shown here, and this is some of the work by Joanna Savilampi in Europe, as she's an anesthesiologist and very interested in particular remifentanil and its effects on airway protection. And in this study, she had subjects drink a fluid that was laced with barium. And she imaged them, right? And they were just sitting upright, and they had actually a non-anesthetic dose of remifentanil on board, right? They were awake and talking. And in the control group, you can see that the contrast is all present in the stomach after drinking that, right? But in the remifentanil group, you can see there's a lot of contrast right in the airways, okay? And these subjects had no idea they had aspirated. And so aspiration can occur without anybody's knowledge or without inducing a cough or anything, but they drank that material, it passed right through the vocal folds, right into the airways. Okay. This can happen to anybody, especially with neurologic diseases. Okay. So all these suite of behaviors, right? They all can break down in various ways and lead to aspiration, which then allows oral and pharyngeal pathogens to pass through the pharynx and on into the trachea, creating a high risk for infection. Okay? And so this is sort of an illustration of the laryngeal adductor reflex. Typically, a speech pathologist or an otolaryngologist will pass a fiber optic scope through the nose into the upper pharynx, where in this area, they can visualize the larynx, right? And so this is the posterior wall of the pharynx, and then here's the epiglottis right here, which during, say, swallowing would fold over and close up the orifice there. And here's what they call the vestibule, but 5 here are the vocal folds, and 6 is the hypopharynx, but also you can see the tracheal rings behind. You can see all the way into the trachea in this image. And you can see up in the upper right here is that same kind of image. Here's the vocal folds, they're open, and here's they're closed. So in this case, one might imagine the laryngeal adductor reflex happening rapidly and closing the vocal folds to reduce the risk of intrusion of material into the trachea. And that's effectively what it does. And here's an image of the muscles that control the vocal folds, which will be here. Here's the thyroarytenoid, which is one of the main muscles that adducts or closes the larynx. But there are other muscles of cricothyroid that contribute to that. And there are also muscles that dilate or abduct, and that would be the posterior cricoarytenoid muscles. But the vocal folds can be positioned, right? They're very important in speech, as you know. And the activity of these muscles as agonists and antagonists defines how rapidly the folds close and whether they're completely opposed or not, right? Whether it's a tight seal or loose or just wide open, for example, when you're breathing. Oops. So LAR is an abbreviation for laryngeal adductor reflex. And I wanted to show this slide to show you this happens in humans. And you can record it, you can actuate it and record it. Here is stimulating the superior laryngeal nerve in a human. You can see the artifact here. And this is a moving average of the electromyogram of the thyroarytenoid muscle. And you can see an early burst, a pause, and a second burst over the course of 100 or so milliseconds. And the first one is R1 or reflex 1. The second was R2. It turns out that the R2 is very susceptible to anesthetic suppression and is likely to be due to suprapontine inputs coming back to affect the excitability of the motor neurons innervating the muscle. R1 is more of a brainstem mediated behavior and happens at short latency here. You get a sharp burst and then a pause, okay? And so it's a really rapid event. And here shows some features of the R1 or the laryngeal adductor reflex R1 in humans. And if you activate it electrically, there's a latency to onset of about 22 or 20 or so milliseconds. It's about 10 to 15 milliseconds latency in a cat, which is our main experimental model. It lasts about 20 milliseconds, so it's pretty brief. You can activate it mechanically by a puff of air or a little von Frey filament applied to the larynx, and it's a longer onset. It lasts longer, and so you can see those numbers here in the human, right? So that the reflex that we're looking at today in our cat model is the R1, and it is important in airway protection in humans. And this is what it looks like in a cat. And these are anesthetized cats that are spontaneously breathing. They're anesthetized with sodium pentobarbital, which is a common anesthetic that is also used in humans. So these are electromyograms of both thyroretinoids. Are bilateral, okay? And so at the arrow, a little von Frey filament was used to touch a vocal fold, and you see a big burst in the electromyogram that lasts quite a while, and meaning hundreds, about 200 milliseconds or so. And then on the contralateral side, there's a burst also, but it is more reduced. And we routinely see this in the cat model. And in panel B, you can see the left and the right bursts, and then also we had the model arranged such that we could measure pressure in the mouth, and then when the vocal folds closed, you can see the pressure rise. We had airflow moving from the mouth through the trachea, right? So when the vocal folds close, they really do occlude the airway and provide a significant barrier to intrusion by materials that are in the pharynx. And so and that was an example of the mechanically induced behavior. So this is a really nice illustration to talk about what's going on in the experiment. So you've got the motor innervation from the larynx comes out the vagal trunk and branches off in the recurrent laryngeal nerve and comes back up along the trachea into the larynx. The sensory information, although there is some sensory in the recurrent laryngeal nerves, most of it is superior, right? And the superior laryngeal nerves can be followed. They join the vagal trunk right around the nodose ganglion. So in our experiments, we simply cut the peripheral end and put the central end on electrodes, okay, for stimulation. On the right side, you can see the current hypothesis for how this reflex is produced. Here's a cross section of the medulla with the cerebellum intact, and of course the sensory input comes in through the vagal trunk up to the nucleus of the tractus solitarius, where the second order interneurons which receive that afferent information are located. And the hypothesis is that information is then transmitted to the motor neurons in the nucleus ambiguous in the ventrolateral brainstem. And then, of course, the axons go out and they're fast conducting to the laryngeal muscles, okay? And then here's sort of a schematic representation of that very thing. So the sensory receptor axons would be exciting second order interneurons in the NTS. And then you'd have an axonal projection to the motor neurons, and there would be synaptic excitation there. And then that information will be transmitted back out to the adductor muscles. So it's thought of as like a two synapse pathway in the brainstem, right? The simplest hypothesis is that the second order interneurons get excited, they excite the motor neurons, and then back out, okay? Of course, the punchline is our data suggests it's a lot more complicated than that, okay? Okay, so far, I've talked to you about electrical activation of the laryngeal afferents, which I'll show you in a moment, mechanical stimulation, and then you can pass aerosols of capsaicin over the larynx, and you'll see laryngeal adductor reflexes, so you can activate it through C fiber activation. There are two components. We're going to focus mainly on R1. It's a bilateral behavior, interestingly enough. And there really is relatively little evidence supporting a detailed knowledge of the circuit in the brainstem. Now, you might wonder, well, okay, why would we really want to know that? What that circuit is like? Well, if you're trying to apply energy, let's say to the vagus, if you've got a device for vagal stimulation, and you're trying to actuate some central system to provide a therapeutic benefit, these central circuits are important because they're the ones that are processing the energy that you're transmitting to the CNS by the nerve, right? And so knowing more about them and how they process that sensory information is important, is an important a piece of knowledge that could inform how you would decide to stimulate the vagus. Okay, and we can talk more about that later. Okay, so here's that accepted hypothesis diagram I just showed you. And here's a modified proposed circuit that we have that probably underestimates what's going on. And it's similar in that here's the laryngeal afferents, in this case through the superior laryngeal nerve, activating second order interneurons. Okay. Now, the catch is if it's beyond that, like another neuron downstream, you would call it third order, fifth order, fourth order. But to put that whole family of neurons together, we just say nth order, right? It turns out that Stuart Mazzoni in Australia, using pseudorabies virus in the rat, has shown that, at least from the lung, the total substrate of interneurons mediating tracheal afferents is probably an order of magnitude larger than the number of neurons that are second order. So this population, just in the NTS, is massive in comparison to this population. So that's a formula for processing and modification of the afferent input, just in the NTS. Okay. Now, these neurons, according to our hypothesis, would be their afferent information would be mediated by other neurons, which then activate what we call expiratory laryngeal motor neurons that innervate the adductor muscles. Okay. The VL for ventrolateral, by the way. That's the ventrolateral brainstem. Now, you might be wondering, what is this ventrolateral oscillatory network? Well, I'll talk about that. But ultimately, to give away what I'm going to tell you is this is a system for tightly regulating the excitability of the motor neurons. Okay, and because we applied, when we electrically stimulated the superior laryngeal nerve, a single pulse at a time, okay, it actually revealed a fingerprint that showed that this was here and allowed us to use that as a tool to understand what the circuit probably looks like. Okay, so here's an arrangement. We anesthetize these cats. We put a trach tube in and allowed them to spontaneously breathe. And then, of course, we put electrodes in the superior laryngeal nerve and recorded electromyograms from most of the respiratory muscles. Here's the adductors, that thyroarytenoid, the posterior cricoarytenoid or abductors that open the larynx. Thyropharyngeus is a pharyngeal constrictor. Thyrohyoid, geniohyoid, myohyoid elevate the larynx so that the epiglottis can fold over the laryngeal orifice during swallowing, right? And of course, there's upper esophageal sphincter that prevents material from, it has to relax to allow material, to go from the pharynx into the esophagus. And I'm not showing it, but when you electrically stimulate the superior laryngeal nerve, the thyroarytenoids are not the only ones that respond. There are weaker responses in many of these muscles. So they all are activated to some extent when you do that. So it's a more widespread behavior than is really related in the literature. Okay. So here is the laryngeal adductor reflex as induced by a single pulse at threshold stimulus currents to the superior laryngeal nerve. And these are probably very fast myelinated afferents that we're activating. And then you see, here's the electromyogram. You can see a sharp burst here at about 15 milliseconds or so. And it's better shown in this moving average where you can see burst, decline. Okay? And you can see it here. And this is not just a random thing. This is most of the animals show this up, down, up, right? And we'll get to this in a little while, like in the next slide, but I'll talk a little bit more about some of the features of this behavior. First, the LAR is subject to frequency depression. Low frequencies have a higher magnitude than higher frequencies of electrical stimulation. And right there, that tells you that the central system is a low-pass filter. Okay? And we've played around with ways in which we can get around this. And we think that instead of using fixed frequency stimuli, you could probably use stochastic patterns of electrical stimulation and sort of defeat the low pass filter to some extent, because that's what's preventing energy from being transmitted from the nerve to the rest of the system, right? You could use high frequencies, but it's not going to follow, and you'll see some of the circuits that are mediating that. Another feature of this is that and a lot of the neurons that participate in this are breathing modulated, but some are not. But if you electrically stimulate during the inspiratory phase of breathing versus the expiratory phase of breathing, stimulation during the E phase is the magnitudes of the LAR are higher, right? These neurons, just during breathing, are normally depolarized during the expiratory phase, so they're more excited. But on the that was the ipsilateral side. But on the contralateral side, that's not the case. So we think the circuits controlling the other side are a little different, and there's other evidence for that. But we're going to focus mostly on the control system for the ipsilateral side. But just keep in mind, there's some interesting features with regard to the bilateral control of this system. So here's some heat maps, okay? And on the left, you can see this is an average of, I think, 100 sweeps of the LAR. And so you can see it's up, down, okay? So these oscillating features are time locked effectively. So so that's why when you do an average, it's not just like one peak. These peaks and troughs get defined because they're temporally linked to the onset of the stimulus, okay? And you can see that in the heat maps. So here's control. And in this animal, we gave actually fentanyl intravenously in increasing doses. And I put this in here because we had an animal where the LER was enhanced, right? So you can see the faded line is where the stimulus starts, okay? And at a lag time, you see, you know, a bright line followed by darkness and a less bright line. So you've got excitation, suppression, excitation. Okay, there's our oscillation there. Okay. But when the LER is enhanced, you can see these waves are there's more of them. So these waves were still there, but subthreshold. Okay, so you have these time-dependent oscillations in here. Okay, now this facilitatory effect was depressed at higher doses and then reversed a little bit by naloxone. And when you look at the power spectral density here, you can see it's very bright here at these low doses, consistent with this oscillation being enhanced. So there's this oscillation of the behavior that's happening that nobody really talks about. But you can actually see it in the human recordings too when you spread them out in time that people have published. And that's one of the features that we're really interested in and what that means. Okay, so how do we study this? Well, we put a bunch of microelectrodes in the brainstem of these anesthetized animals, okay, and recorded many neurons simultaneously, both in the NTS and other areas. And in the left, you can see some of the signals we get. Each of these vertical spikes is an action potential from a neuron, and some of the neurons are modulated during breathing. That is, when you record the phrenic nerve that innervates the diaphragm, some neurons are active only during the inspiratory phase, coincident with the phrenic nerve, or just during the expiratory phase when inspiratory activity was not present in the phrenic nerve, right? And then we can sort them, and then you come up with these TTL pulses based upon the occurrence of these spikes. Each pulse is the occurrence of a single spike in the neurons, okay? And then I for inspiratory, E for expiratory, AUG for augmenting, E for decrementing based upon their major pattern, okay? That's less important than just to introduce what we did. On the right side, here's a cat skull for reference, and this stereotaxic apparatus, right? And you can see these devices here. These cylinders are worm drives, and they're attached to long tungsten microelectrodes, and I'm not sure if it comes up on your screen, these long electrodes. And then we have these plexiglass manifolds that make that bring down the tips of these microelectrodes to about 300 microns apart. And, you know, at USF especially, they could put as many as 100 electrodes in the brainstem of a cat, but we're only up to about 40 or so in my lab. It's not a lot of room there. But the point being is we can record from a lot of these neurons simultaneously, which is important in trying to understand how this system functions as a network. Here's some examples of what the responses of these neurons when you stimulate the superior laryngeal nerve, right? So at the bottom, here's an SLN stimulus right here, superior laryngeal nerve, and then here's the recurrent laryngeal nerve motor axon response. You can see here's this adductor reflex that happens after about 8 milliseconds of this out. And here's just some in this case post-stimulus time histograms showing before and after the stimulus occurrence. And you can see that some neurons are pretty fast here. They're a little bit faster than the behavior itself. Responders, some are lagging, and some are active really pretty much after the behavior is mostly happening. At the bottom, we did spike-triggered averaging of the vagus nerve and showed that this neuron was in fact a motor neuron. And then based upon other analyses, we think this was a premotor neuron, meaning it's exciting the motor neuron. And then, but you can see that some neurons are peaking sort of after the motor neuron, some a little bit before, right? So you get this temporally distributed array of responses of these neurons throughout the behavior. Okay, so that's the layout of what the behavior looks like, some features we're interested in, and then how we tried to study this. But now we're on to the issue of, okay, how do you study a network? Okay, and so our approach has been to record the activity patterns of these neurons and compare the times of occurrence of the spikes relative to each other, each neuron. And in doing that, you're conducting what we call cross-correlation analysis. And in doing that, you can obtain information on whether you can use to conclude whether neuron A excites neuron B or neuron A inhibits neuron B. Okay, and here's some examples of that. And in these cross-correlation histograms, the zero time is the occurrence of a spike in what we call the reference neuron. You got to start somewhere, right? In this case, 806 and 828. Here's 806 in our model and 828. Okay, and so this faded line shows the accumulated the onset of a spike in 806, time before that and time after that of the activity of 828. Okay, so a spike occurs in 806. What happens after that spike in 828, and what happens before? Okay, so after a spike in 806, you can see an increase in counts, accumulated counts of spikes in 828, consistent with excitation. And so the hypothesis is 806 excites 828, shown by this sort of synaptic symbol of this white. Okay, and that's a high resolution record. This is the same recording at more compressed in time. Okay, and here's a separate recording between 830 and 806, 830 and 806. And so by the same token, when a spike in 830 happens, there's actually a decrease in probability that 806 will fire, consistent with 830 inhibiting 806, shown here by a closed sort of synaptic symbol. And that's how you kind of put a network together sequentially. And in this case, these are serial functions, like a bucket brigade, okay? A to B to C to D. And we think of these circuits as being serial functions, right? Almost exclusively. In the reality, that's only about half of what happens, okay? The other half of what happens are parallel functions, okay? And those are shown here with these same correlation histograms. If you take 415 and 727, right? At the zero time, a spike in 415 happens, and then you're looking at the activity pattern of 727 in response. But there's a peak that's mostly central. Same for this pair, same for this pair, same for this pair. So basically what that's showing is that 415 and 727 and these other ones are discharging at about the same time. So it isn't really consistent with cause and effect between those two neurons. It's more consistent with something that looks like this. There's a common neuron that you're not observing that is exciting them both. And so they have an activity pattern that's about the same time. So that's a parallel function. And like I said, we have a lot of background in this area in the brainstem networks. Parallel functions or mixed functions involving a parallel function are about half of what we see. And nobody talks about this kind of stuff. They're an important feature of how networks retain excitability. And they're also mixed functions, where in this is a model that was run by our collaborator, now retired Bruce Lindsay, where he set up in a simulator a neuron that drove two other neurons, but one of the other ones inhibited the other one downstream. So here's our parallel function right here, and here's a serial function here. And how would that cross correlation histogram look in simulation? Well, you'd have in this, a peak that was offset a little bit from here, and then a trough, right? So both neurons are excited, and then 2 exerts its inhibitory effects on 3. So if you're looking at 1 and 3, you're seeing a peak and then a trough. I call this a limiting function. Something like driving a car with your foot on the gas and foot on the brake at the same time. It's limiting the excitability of one of the targets. It's excited, but then shut down, okay? And that's about 30% of what we see in these networks, these limiting functions, okay? So that's sort of the backdrop of how we put a network together and how we kind of decide how neurons interact, is the features on these cross-correlation histograms, how we interpret them, okay? Okay, so here's the bread and butter of how we're trying to figure out how these sort of oscillatory features in the laryngeal adductor reflex happen. Okay, here's our model prediction again, and here's our averaged EMG with the peak trough, right? And then here's our raw record. And then so we have 2 neurons here. One is an expiratory laryngeal motor neuron that we recorded. And notice it's got a peak, trough, right? Just like the EMG, as you'd expect, okay? And so there was another neuron we recorded, and this neuron was active mostly during breathing, but it was recruited when we electrically stimulated the superior laryngeal nerve. So these are 2 peristimulus time histograms. At the faded center line was the occurrence of SLN stimulus, a single pulse, 100 microseconds. And then you see the time before and then the time after. And so at short latency, we see this neuron actually turns on a little bit earlier, it's harder to see here, than the motor neuron. But interestingly enough, it has this interesting pattern where it spikes really, it has a high frequency of spikes here, right when the motor neuron is depressed here. And when we ran the cross correlation, we observed a central peak, indicating these neurons are firing at about the same time on a short time scale basis, which is consistent with the fact that we're both excited by superior laryngeal nerve stimulation, but also there was an offset trough consistent with this neuron inhibiting the motor neuron. And notice it has a very high level of activity when the motor neuron had a low level of activity, okay? And so what we think is happening is in this example is this is sort of the grist for the mill and how these oscillations happen. These are very sharply time-dependent synaptic processes for activation and inhibition that create this oscillation, right? The motor neuron turns on, but then it's inhibited by a neuron that comes on at about the same time and spikes then. When this falls off, then that allows this second peak to happen. And then see, there's another peak later, okay? So it's these rapid, really strong synaptic effects between these neurons that create this oscillation. That's the evidence that we have so far. So there's other sorts of features, other sorts of control mechanisms which we observed, right? In the left, we also happen to record from neurons in the medial reticular formation that were on the same rostral caudal level as the facial nucleus, but they were quite medial. And that would be MRF here. And here's another neuron, which we called 808 in the ventral respiratory column, the ventrolateral area. And when we cross-correlated these two, it turns out that 434 was inhibiting 808. But they both were excited by electrical stimulation of the superior laryngeal nerve. But there was a temporal offset between the peaks, okay? So it turns out that 808 has this really sharp onset peak, but it comes back down pretty rapidly. That again is not an accident. Usually, if the rate of rise and the rate of decrement of these peaks are the same, there's some pretty important regulatory issues controlling that. Now, it turns out this guy turns on a little bit later. And since it's inhibiting him, we think that's contributing to keeping this guy low, this activity pattern low, and this decline. It's more apparent here for this pair. And here's 434 inhibiting 808. Okay. I'm not showing the cross correlation histogram for 406 and 805, but here 805 has a really sharp, very short response to electrical stimulation of the superior laryngeal nerve. We think in part because this guy is inhibiting him and he comes on later. So this guy is inhibiting this guy and he's contributing to the fact that it's silent after that really sharp onset. Okay, and that's a fingerprint of really a strong synaptic control over the excitability of the neurons that participate in these behaviors. Okay, we also think there are other control mechanisms, including what I call feed-forward excitation. And so here's a ventrolateral brainstem neuron, the ventral respiratory column, which we designate 821. It was excited by electrical stimulation of the superior laryngeal nerve at short latency. And this is it, right? Here's the PSD histogram. Short latency, and you can see it actually has these oscillations in it. Okay? Very sharp onset, very short. By cross correlation, 821 excites 709 and 715, and both of these neurons are in the NTS. And you can see their peaks actually lag the peak of 821. So we think that 821 is contributing to the excitatory responses of these neurons, except that in a cross-section of the brainstem, including the NTS, in our schematic model, this would be 821. And then there would be second-order interneurons, which we didn't record from, which would be exciting 821, and it is feeding back to the NTS and exciting other neurons later than the second orders. So that's an example of what we call feed-forward excitation. Okay, so yeah, the second orders are excited. They excite other neurons out here, but they do feed back and create a situation where you now have kind of this this pattern where there's you need kind of both areas to excite the whole behavior such that you can get an orderly behavior produced in the motor neurons. Okay. Okay. Excuse me. So here's a circuit that we put together from one animal. Okay. And on the left, here's a stylized brainstem with the cerebellum removed and a dorsal view. Here's the fourth ventricle, and this is rostral and this is caudal. Okay. So here's the nucleus of the tractus solitarius, the raphe nuclei, the ventrolateral areas, and the pons, where the areas which we recorded from. Okay. And on the right side, we pieced together a circuit. I put this up here because it's got two expiratory laryngeal motor neurons that innervate the adductors. Okay. And this would be in the ventrolateral network and the nucleus ambiguous, where this part of the circuit would be located. Here's medial reticular formation, pons, raphe, and the NTS area. And so the superior laryngeal nerve afferents are coming in to the NTS, and then you can see a host of things happening. Here's this neuron. There's a lot of acronyms here. Here's non-breathing modulated. This is a neuron that's active during the inspiratory phase of breathing. And this is an expiratory neuron, T for tonic, is sort of continuously active. Right? And so you can see here, for example, the inspiratory neuron gets feedback from the motor neuron. And then blue is excitatory, red is inhibitory. Okay, so it's being both excited and inhibited by this guy. And that is consistent with the limiting function, right? You're exciting, but you're inhibiting as well. Foot in the gas, foot in the brake. Okay, and this neuron is inhibiting this neuron, which is exciting this neuron and inhibiting this, right? So you get these sequential serial functions, which actually go all over the place, okay? But what's important to know here is that these other areas are involved too in controlling the excitability of these motor neurons, and the motor neurons do other things to these areas themselves, okay? So this is an example of a control system that is way more complicated than the two synapse control system that people thought. And for such a simple behavior, it's really interesting to see a really complex circuit like this. And then you can imagine it would be hard to predict exactly how energy would be transmitted through this circuit if you were stimulating sensory afferents from a visceral organ that travel in the vagus nerve, right? And so to me, I'm always surprised that you can do anything to a nerve and get something that is therapeutic happen, right? Because of these circuits that are just crazy complicated, right? So, but anyway, some of these mechanisms contribute to these oscillations. For example, here's an inspiratory neuron that is both exciting and inhibiting the laryngeal motor neuron. The real question is, and I don't have on here, is what is the time lag of this excitation and inhibition? It can be the excitation happens first, the inhibition second, and so you get a burst and then a pause or a trough, like that oscillation. Okay, so we think that some of these synaptic effects are really strong, and then when they're activated by a single pulse in the LAR, they create a time-dependent cascade through the network, which allows us to detect this control system that is manifest as oscillations. But what it really represents is the production of a behavior that is under significant inhibitory control. And it's a strong gain control mechanism of this behavior. Okay. Okay. So to close, so the superior laryngeal nerve through probably fast A fibers activates the adductor reflex, and this behavior is important in preventing material from penetrating into the lungs. The hypothesis that people have had for a long time for the nature of the circuit, central circuit that controls this behavior, is an underestimate by a long shot of what the circuit looks like. And you can see these oscillations in the circuit that they're a fingerprint of the of powerful synaptic effects within the medulla that regulate the excitability of the motor neurons, so that these behaviors can be initiated and completed in a reasonable amount of time to then move the vocal folds to do the right thing according to the environmental conditions the afferents are exposed to. So thanks very much for listening and the opportunity to present my work. So
Thanks, Don. I find that super fascinating, and I've got a bunch of questions.
oh wow.
But
Okay.
but I want to open the floor for everyone else to pop any questions into the question panel. You may also see a survey pop up. Feel free to type your questions first and then complete the survey if you so desire. But while we wait for folks to contribute some questions, I'll ask something that has sort of piqued my interest, which is how do you coordinate between left and right? Because obviously you want everything to happen synchronously, right? Equal and effective to have both vocal cords close at the same time. And
Right.
is that what that oscillation is for? It's to help
Well,
it
the
train the
yeah, the oscillation, when you stimulate just one side, the oscillation on the ipsilateral side isn't nearly as profound. And then
Mhm.
there are hypotheses about crossing pathways through the reticular formation, right? But rarely is it that there's sensory stimulus, like some particulates might be exposed to only one side of the larynx, right, and not the other side. So, you know, it's likely that both sides are coordinated based upon the nature and distribution of the stimulus across the afferent system to produce an organized behavior where both sides adopt at the same rate. But we have looked at opioids and the right and left side. It turns out the contralateral side is more sensitive to opioids than the ipsilateral side, and which is consistent with a control system. We've also microinjected codeine into the contralateral side to see if it acts directly there to suppress the behavior. And it turns out that when we do that, we actually suppress the other side, so that there's that in the region of where the motor neurons are supposed to be, when we microinject codeine, they're crossing pathways to go back to the other side. So there's that are codeine sensitive. So there are intramedullary pathways that connect both the control systems for both motor neuron pools. If that's answering your question.
So, so that's really fascinating. Okay. So opioids can produce a stronger contralateral response than ipsilateral, is what you're saying?
Yeah, that's something we've seen.
Holy moly.
Yeah. But generally, the laryngeal adductor reflex is the ipsilateral side is pretty resilient to opioid depression compared to, say, cough reflex. And I've been, just amazed at how resilient it can be. The contralateral side gets, knocked away, but the ipsilateral response is still pretty strong.
And that's driven by the afferent.
Well, I think that a collaborator of mine, believes that their mu-opioid receptors do not exist on the laryngeal motor neurons, but they could in the premotor pathways, for all we know. And
right.
but that's just the manifestation of what we're seeing. And so what we do know about the effects of opioids in the brainstem network is that they're not just depressants. You see some neurons increase activity with opioids, others are depressed. So it's really, the way I think about it is a balance of excitability, right? And then to use a personal example, when my son was a teenager, we had a trampoline, and he and I would jump on the trampoline, and his friend next door was like 250 pounds. We get on there, and then the balance of levels where we were would change a lot when he climbed on the trampoline, right? The trampoline could be considered a network, right?
To electrical conversion there. So we have a couple of questions that have popped in. And the first is a question. And the question is, do you think that the complexity is needed to manage the variability and other demands on the airway?
Absolutely. And because the vocal folds, obviously they're involved in speech, which is a very precisely regulated behavior. And they're involved in swallowing, right? Cough, a behavior known as the expiration reflex, laryngeal elevation, all sorts of things. And so you've got this multiplicity of different behaviors, each of which is regulated differently. So you need a pretty complicated network to manage that. And then the speech pathologists study all sorts of laryngeal behaviors and pathologies, many of which are manifest as a change in the ability of the vocal folds to medialize when they're supposed to close, right? Some don't medialize as much. Like one side doesn't medialize as much as it should, right? So I think the other thing is the way the muscles are arranged. You can take an analogy from the skeletal motor system, the appendicular motor system, meaning that when you have a joint, people talk about joint stiffness when you lift something. And so the idea, it's not just one muscle that's controlling the movement across that joint, it's the antagonist also stiffening it, right? And so that's probably analogous to what's going on with the vocal folds. It's not like when the vocal folds adduct some that the abductors are silent. They can be resisting that a little bit, and it creates a system where you're controlling the position of the vocal folds depending upon the needs. So yeah, it's going to be a very complicated system because those motor neurons have a lot coming in, moment by moment.
Right, right. But they also need to be very resilient to,
Right.
um, mhm, got it. Okay, so we have an alternative uh, uh, comment here, looking for your feedback.
Okay.
So the comment is, I would like to propose an alternative but not mutually exclusive mechanism for the observed low-pass filter effect during
Okay.
superior laryngeal nerve stimulation. Since laryngeal adductors contain fast twitch fibers, they're
Yep.
susceptible to fatigue at
Ah,
frequencies over 20 hertz. So what do you think?
yeah. Fatigue would be something that you should see evidence of the longer you stimulate,
Hmm.
right? And I think that that's a great question. And we haven't really specifically analyzed the features of that behavior. For example, that oscillatory behavior at uh, at uh, at 10 seconds versus a minute of continuous simulation versus 5 minutes of continuous simulation, right? That would be the way to know to see if there's a time dependent effect in those oscillations. That's a great question. We haven't looked at that.
Oh, okay. Anybody have any last questions? Go ahead and pop them into the um, Q&A panel if you'd like. I found this incredibly fascinating. The idea of such a complex series of neuronal circuitry within the NTS that is sequentially ordered and yet parallel and serial at the same time is really fascinating to me. But I want to thank you, Don, for taking us through this laryngeal adductor reflex. Can you tell me one more time, just remind me how clinicians use the laryngeal adductor reflex and what it's indicative of? I know you
Yeah,
covered
I
it
mean,
at the
like I said, they can scope a subject and have a little air puff
beginning.
on the epiglottis, the vocal folds, and that is a stimulus to understand how effectively both vocal folds medialize, and whether one is slower or one over medializes. And sometimes there are peripheral nerve injuries, and sometimes there's central issues, right? And so it's a very common um, intervention to sort of see, okay, how is that happening, right, through reflex. And
Right.
so that pattern you can visualize the rate of movement, and a clinician can get a sense of whether the motor innervation is competent on both sides or not.
Right. And that leads into assessment of risk for aspiration and other types
Well,
of.
that and then there, for example, RFK Jr. has spasmodic dysphonia, if you hear him talk,
Mhm.
and they'll inject, Botox, and that uh, usually relieves that phenomenon, that condition for a few months. But that's sort of one thing they might do, and sometimes there are neurologic conditions affecting the vocal folds, right? The control of the vocal folds, and so that's
Mhm.
how one way they diagnose some of these things.
Gotcha. All right. Well, Don, I want to thank you for your time and audience. Thanks so much for being here to hear this great seminar. I really appreciate it. And the interactions of this really complex central feedback mechanism is really pretty phenomenal and clearly complicated. The rig that you had demonstrates just how incredibly massive that is. And also the impact of being able to record from all those neurons and then feed it into a computational simulation then allows you to tease out all those different pairwise comparisons, right?
Yeah, I mean, the model approach is really, I haven't shown it, but we have a network model that produces breathing, coughing, now swallowing, and we're trying to introduce our laryngeal adductor reflex circuits into it. But that all that does is it tells you the plausibility of your circuit, right?
Right, right.
Does it plausibly produce the behavior and way you think? And then it's like any model, they always say that the models are always wrong, but some are useful. And
Right.
so it's a useful model. And it's sort of like I have a collaborator that says this is the world's most addictive video game, because what you're doing is you're producing motor outputs that look like an in vivo recording of all these muscles and all the neurons.
Hmm.
And I actually, as a quick aside, I years ago I went to a cough meeting and the room was full of clinicians who see chronic cough all the time, and I had run a simulation and gotten the model to produce a cough, right, breathing and a cough. And I showed them that. I was talking about it, and I didn't tell them it was a simulation until at the very end, and then it was like a deep intake of breath because they thought it was in vivo data,
Hey,
right?
that's
They didn't know it was a
cool.
simulation. Now they're not used to
Right, because
looking
your
at those
simulation
signals.
is informed by previous in vivo data, right?
Yeah, yeah. And so the circuits to produce all those different behaviors in one circuit, it gets pretty involved. And then it's all always like whack-a-mole trying to balance it. But that's, I could spend the rest of my career just modeling. Yeah. So
Yeah. Well,
there's not enough hands to do all that work.
exactly. So I look forward to an update from you. Let me know as that model gets more and more complex. I really appreciate your time.
Great. Well, thank you very much for the invitation again.
Yeah. Take care, everybody. Bye, Don.
Thank you for coming.